Hello, welcome to 13 Universal. This is A-level CIE Biology with shared role. So we continue on all topics, paper 4, series 5. Follow us, go through series 1, 2, 3, and 4. Solve the questions with us. Remember, guys, how we do this. We solve questions together, all right? So get your writing materials and let's solve some questions, okay? Remember that the most important skills that you have to recall what you understand and apply it here. Okay, look at the first question, guys. Here, let's roll. It says one cause of genetic disease, severe combined immunodeficiency syndrome, that's SCID, is a mutation in the ADA gene. This mutation results in a deficiency of the enzyme adenosine deaminase. So let's go through the context. Background information is very important. Just follow them. Although ADA is found throughout the body, it is especially active in lymphocytes. The absence of functional ADA causes the build-up of toxic metabolites that kill lymphocytes and damage organs. So you see the impact of this. Kills lymphocytes and then damage organs, right? So if you look at this, babies are often diagnosed with SCID by 6 months old. This is terrible, man. It's terrible. Right, treatment can, okay, thank God there's a treatment. Treatment can greatly improve the life expectancy of children with SCID. Okay, let's see how it goes. Okay, some treatment options are available. Let's explore them. Enzymes replacement therapy with recombinant human ADA made by genetically modified GM Escherichia coli. Weekly intramuscular injections are given, so you keep giving this guy injection. Bone marrow transplant. Okay, if a well-matched donor such as close relative can be found, if the person can be found. Gene therapy. Now guys, read this background information and understand it. It's very, very important. Then the question says suggest and explain why it may be more appropriate to use enzyme replacement therapy, all right, to that of SCID instead of bone marrow transplant. All right, pause this video, go over the question and try to solve this question. This is about 4 marks question. Okay, try to solve this question. All right, so we're trying to suggest and explain why it may be more appropriate. Suggest, okay, make a valid point and give reasons. All right, so we're suggesting that they say why is more appropriate to use enzyme replacement than the bone marrow. Yeah, it's more appropriate, guys, because it is immediate. All you just need to do is inject this person with the enzyme. It is immediate. Okay, yeah, that's number one. Number one reason, apart from the fact that it is immediate. Number two, it should be cheap. It's cheap, and you don't have to look for donor, so you don't have to wait long for donor. Is that okay? And I think it should be less invasive. Less invasive in the sense that in bone marrow they have to extract somebody's bone marrow transplant, and of course when you do bone marrow transplant, it causes immune response. So this may not trigger immune response. I mean, injection of enzymes, it may not trigger immune response. All right, okay, so let's look at what the mass game says quickly. So donor not needed, see. So this, anytime they ask suggest questions, think outside the box. These things are not in your textbooks. Your teacher might have not taught you this, but this is ability to think outside the box. They are just trying to test your ability to synthesize information. Okay, think outside the box. So donor not needed. Immediate effect, yes, it's immediate. So easy to administer, no immune response, less invasive. Okay, all right, low risk of health, cheaper, quicker. Oh, that's it, guys. Let's go to the next question quickly. Outline the procedure used for gene therapy treatment of a person with SCID. So for gene therapy with SCID, guys, don't forget that the first thing you do with SCID is that you just have to extract the stem cells. Okay, so you give the person drug to extract, all right, because to have more stem cells produced, and then you extract the stem cell from the bone marrow, and then you, of course, you must have added the normal allele into adeno-associated virus. Okay, you must have added it, right? so into adeno-associated virus, because you use adeno-associated virus to transfer the normal allele into stem cell. That's gene therapy. So when you extract the stem cell, you now mix it with adeno-associated virus carrying the normal allele, and then the gene is transferred. When the virus gets into the adeno-associated virus, when the adeno-associated virus, which is acting as a vector, gets into the stem cell, right, and then the gene is being expressed. Remember, they say outline. Outline means this is the main point. The main point is that you inject the person with medication to stimulate the production of more stem cells, and then the stem cells are extracted from the bone marrow. Stem cells, are mixed with adenoviruses, okay, that contain the normal allele that makes ADA, okay, and then of course that means it is transduced with this virus. The virus, when it gets in, and then the stem cell is now injected into the bloodstream. Okay, when it gets into the bloodstream, start dividing, multiplying, and then make lymphocytes which can start expressing this gene. So look at it here, guys. So memorize it. Look at it. Give a drug to increase the number of stem cells. Memorize it. Anytime they ask you for this, they will ask you again. Procedure. So give a drug to increase the number of stem cells in the bone marrow. All right. A reference to virus or vector containing normal healthy allele. So you must have, all right, transferred the healthy or normal allele to this virus, adeno-associated virus, then remove bone marrow stem cells, mix stem cells with viral vector to allow transfer, and then radiotherapy, all right, to make bone space in bone marrow, all right, to kill stem cells in bone marrow to create space, all right. So transduced or infused, injected into the blood, the lymphocyte produce this. All right, see, gamma ray, you can use either gamma or retrovirus, this gamma or retrovirus or adeno-associated viruses, okay. Example, tissue from bone marrow, purified, sorted. All right, so please don't forget the procedure. All right, so next question. So just the social and ethical implication of gene therapy for skin that need to be considered before treatment is considered. All right, this is talking about both advantages and disadvantages, social and ethical. Now don't forget that this is quite expensive. Yeah, it's expensive. I hope you know that. So you have to know that it's expensive. Apart from the fact that it is expensive, you also have to know that it is also risky, because you are taking the there's a lot of risk associated to it. You're taking the bone marrow of somebody, okay. So it's risky, and there are a lot of ethical objections. A lot of ethical objections are involved, okay. So a lot of religious belief, people do not, these are ethical issues, people may object it, okay. So what else do you think? All right, so let's look at what the mass scheme says. It said it's expensive. Yes, remember that once it's treated, you don't have to give the person drug again. No longer chronic, only one single treatment. All right, so no donor needed, like we said before. So more money available to health system may cause cancer. So these stressful, these are the disadvantages. Remember, when they say ethical implication, they must mention disadvantages and advantages. Expensive, okay, is a disadvantage, but this is an advantage long term. No need for regular injections, okay. Only a single treatment. All right, these are also other implications. Cultural beliefs. All right, people say you play God. All right, and then people don't believe in gene therapy. All right, okay. So let's go to the next question, guys. A freshwater species, Oryzia latipia, has individual with 4 body color patterns as shown in Fig 5.1. So come look at these beautiful fishes, okay. So these are the phenotype. This is red, white, red with black, okay, white with black spots, okay. So these are phenotypes. So let's look at background information. 2 unlinked, these are unlinked genes determine the body color pattern shown in table 5.1. One gene controls whether the body color is red or white. So dominant allele is red, okay, recessive is white. The other controls whether black spots are present or not present. Dominant is B, which is black spot, recessive without black spot. A fish that is homozygous recessive at both loci is white. Okay, that means it has this without black and white, so that will be white. No black spot, it's not red. Okay, so genetic crosses were carried out to investigate the inheritance of the four different body color patterns. Males that were red with black spots are homozygous at loci. These are homozygous dominant. Were crossed with female that were white. Okay, that's homozygous recessive. The F1 offspring were all red with black spot. Okay, this is a pure breeding experiment. Crossing homozygous recessive against homozygous dominant. Okay, the generation, the offspring produced is always called F1. F1, is that okay? And then what we have here, let's see what we have. Table 5.2 shows the observed numbers obtained of each of the four different phenotypes for F2 generations. So look at F2 generation, guys. Yep, see that? Okay, it says table 5.2 compares the observed number with the numbers that would be expected in F2 generations for a normal dihybrid ratio. Calculate chi-square for generation by computing table 5.2. The formula is, so give me the formula. All right, look at it, guys. So they want you to complete this. So what do you do here? Okay, so what you do here is observe minus expected. So get your calculator, fill up this. I want you to work on that. So observe is 279. That is this. That's observed because this is, we're going to fill this minus E. E is minus 281.25. Good. So that gives you minus 2.25. Yeah, minus 2.25 here. Okay. So that will be minus 2.25. So yeah, so what are you going to do with that? You square it. So that's minus 2.25. Okay. So now this says you should square it. So you square it. So of course, you know, it's gonna give you a positive sign. All right, a positive sign. So if you say minus 2.25 squared, it will give you plus what? It will give you 5.0625. All right, so write it out. I wanted to fill it. 5 point. Let's not waste time. Okay, but I'm showing how to do it. 5 point. Oh, that's 5. Let me clean that. Yeah. So 5 point what, guys? 5 point. I hope you're working on this. 5.0625, right? So the next thing is divided by expected. Yeah. So divided by the expected. Okay. Expected is 281.25. So get your calculator again. That divided by 281.25. What does it give you? All right, it gives you 0.018. So this gives you 0.018. So okay, I hope you're working on this. So I've gotten for this 18. So now we'll do for this also. For the white. So let's go for the white here. This, right? Expected minus, okay. So let's punch the calculator. 30 minus 31.25. 31.25. What does it give you? Minus 1.25. Okay, so that gives you minus 1.25 minus, okay, 1.25. That's a mistake. Let's clean it. Minus 1.25, that's point there. Minus 1.25. Yeah. All right, so you square it. So you bring your calculator and you say, okay, minus 1.25 squared gives you what? It gives you, all right, 1.5, yeah, 1.5625. Okay, 25. That's 25. Okay, so to get this, add up everything. You're gonna have to add up, yeah, this plus this plus this plus this. That's okay. Get your calculator. So 1.625 plus 0.054 plus 0.017 plus 0.018. What does it give you? Okay, it gives us, of course, you know what it gives us. 1 point, yeah, that gives you 1 point, okay, 65, 15. Yep, is that correct? Okay, so if we do it well, we should get something like this. Okay, so that's 1.6515. Okay, now that's the formula we're going to use. So of course we've done that. So you're going to add up everything here. Okay, that's it. Okay, so let's look at what we have there. Oh, there's a mistake there. We got 0.0. Okay, we didn't add it very well, but we got this. Yeah, so we got this. Square this gives us this. Oh, I didn't divide. Okay, I did not divide. It was here we made the mistake. I didn't divide with 3.72. Yeah, so if I divided with 3.725, yes, we would have something like this. Okay, so this divided by this will give us this. That's the mistake. So this is what we should have, guys. Correct it, please. We should have this. So this is supposed to be divided by this because that is observed minus expected all squared divided by expected. So divide by expected. You have this. So 0.14 approximately. Okay, so let's go. Let's say the critical value at P2 is at P 0.5 and 3 degree of freedom is 7.82. 7.815, sorry. Comment on whether the null hypothesis should be accepted or rejected. So how do you make this decision, guys? Okay, remember how we go about it. You see, we have two chi-square values. We have chi-square, okay, chi-square calculated. So this is it. The one we just did now is the calculated value. If it is greater than chi-square table value, is chi-square table value, all right? Chi-square tabulated. Now, if what we calculated is greater than the table value at critical probability 0.05, what you just do is that you reject the null hypothesis. Okay, so remember that what we have here is 7.85. Our calculated is 0.14. Our calculated is just 0. Okay, 14. 0.14. That's 0.14, while our tabulated is 7.8. 7.815. Can you see that 0.14 is not greater? Remember that the condition is the short making condition, that if chi square calculated is greater than chi square tabulated, reject the null hypothesis. Null hypothesis says there is no significant difference, right? But in this case now, our calculated value, which is 0.14, is not greater than 7.85. So we accept the null hypothesis. So you understand? So null hypothesis says there is significant difference. Do you get? So there's significant difference. So what we're going to say is that there's this significant difference. All right? No. Null hypothesis says there's no significant difference. Yes. So it's null hypothesis. It states that there is no significant difference between the expected and the observed. So look at it. So chi square value is lower than the critical value. Okay. So the observation results, they are not significant different, all right, to the expected number. So once, remember that null hypothesis always says there's no significant difference between the expected, and that's what we're going to accept here. Once calculated is greater than this, reject it, but if it is less than this, accept it. So we're accepting that there's no significant difference between the expected number and the observed number. Okay, so any difference are due to difference there is due to chance. All right, so let's go to the next one. So further analysis of the result from F2 generation in table 5.1 showed that there were no white males or white males with black spot. In O latifs, females have 2 X chromosomes and male have X and Y, just like in human beings. It was deduced that in O latifs, the gene that controls body color is located on X chromosomes and Y chromosomes. Okay, the genes that controls whether black spot or not, right, is located on an autosome. Autosome is non-sex characteristics, non-sex chromosomes that carry non-sexual characteristics. That's autosomes. We have sex chromosomes, they carry genes with sexual characteristics, right? So look at this. So it says to produce, look at that, to produce the F2 generation, the red males, all right, with black spot, this one, okay, yeah, was crossed with red females with black spot. That complete the Punnett square in Fig 5.1 to show the genotype and phenotype of the F2 generation. Use symbols, you can see that. So use symbols, this for the alleles, for the genes that control body color. Use symbols, this for the one that controls black spot, right? Okay, so let's look at it. Some of the Fig 5.1 has been completed for you. All right, so let's look at it, guys. That's it. So that's how we should complete it. So pause this video and try it out. First of all, try it out. Okay, so if you want to write, okay, so first of all, you match it. We're going to fill in here. So match this and this. So for this one, match this and this, match that and that to get here. Okay, so guys, now we're just going to do one. So we're going to match this and then show you how the rest was done. So that's going to be, okay, X, that's a female, right? X, okay, that's red color. Another X, superscript small r, okay, that shows red color because red is dominant. So we have the black guy is dominant. You have this, okay, and then yeah, so homozygous for black. So this is a female, right? Is a female, okay, and this female is red, all right, with black spot. So female with red black spot, okay, red body color and black spot, okay, yeah, so that's female. So it's going to be red and black spot, red and black spot, right? Black spot. So that's it, guys. Now black spot, you can write it down. Now the same thing. Look at this one. this against this. This is going to give us female also, but it's going to be white because that's recessive. White with no spot, so that's white. This is white. Okay, so if you compare this and this, okay, this one and this is going to be male. Okay, red color with black spot. All right, this and this, male, red color and no spot. I would have done this. Okay, this and this is going to be male. All right, it's going to be red and no, male first, red and no spot. Why is it male? Because it's XY. All right, let's look at what the math scheme says, not wasting time. So you can see that. See all the ones we did. Female. Okay, so if you say this and this, right, this is, if you combine this and this is white. Okay, and this is black spot. Okay, and look at this one. If you combine this and this is female, right, and no spot. Combine this and of course this is going to be red, male and black spot. Okay, red, male. Combine this and that, red, male and no spot. Okay, so we crossed the wrong one, but it's supposed to be X superscript. Okay, small or lowercase, all right, and X superscript lowercase. Okay, that's going to give us white, and because this is present, this is black spot. Okay, all right, so let's move to the next question quickly. Explain why there are no white males or males that are white with black spot in F generation. Now there are no white males because number one, we do not have Y gametes, all right, that has white allele. White allele is this superscript. We don't have it, right? It's not present. Okay, so this is not present. This is not present. It's only the dominant that is present. The dominant allele is this. Okay, superscript that's present. So that's the one that was passed on. Okay, so yeah, that's it there. So allele R dominant is on Y chromosome, so all males inherited dominant red. Okay, so that's the dominant red. It was the one present, so they all inherited this. Okay, so they do not inherit, or I can say that this is present in the gamete. No alleles recessive. Okay, all right, alleles only exist on X chromosome. Now even the X chromosomes, they have only the, okay, the recessive form. So males never inherit recessive white allele. That's it. Okay, there's another cross, red males with genotype that were mated with white males with genotype. Okay, that now this is white, no spot. This is white. No, this is red male, no spot. Cross with white male, no spot there. All male offspring were expected to be red, and all female were expected to be white. Okay, the observed results show that the offspring included 2 red females out of 253 and 1 white male out of 198. So just an explanation of this unexpected result. So guys, you see, we're not, if you cross this, we're not expected to have red female, because look at the X chromosomes here, they all have white allele. Look at it. Okay, so but in the result, red female showed up. We're not expected to have of course white male, because the dominant one is present. This is dominant red. Okay, even though we have white allele for male on Y axis, on Y chromosome present, but because of the dominant, we're not supposed to have white male, because this is going to dominate. It's going to, all right, mask this. However, it occurs in the result. Okay, why does it occur in the result? The reason is because of maybe mutation has occurred. Okay, So mutation occurs, and of course mutation could be, base substitution, base deletion, all right, occurs on the gene. And because mutation occurs, instead of having red, okay, instead of having white female, we're having red. The same thing here. Or it could be as a result of crossing over, guys. Yes, crossing over. There's, during metaprophysis 1, crossing over could occur. So if I have a crossing over here, there's a gene here. So these are two. I have Y. I have, this is, okay. And I have of course, okay, crossing over occurs, right, between two major chromosomes, okay, but they have to be homologous. You get, maybe XX or whatever, but we know that, or even Y and X, crossing over could occur, and they swap places. So if crossing over occurs between non-sister, all right, chromatids, this has white here. So imagine, guys, okay, this swap places, this comes here and that comes here, okay. All right, it happens between an X chromosome and Y chromosome, okay. That is the explanation here. All right, so look at the last question here. It says fig 6.1 shows part of the Bowman capsule of a kidney. Look at this. Name structures A, B, and C. So pause the video and try it. So A is endothelial cells of capillary, endothelial cells. You see that B is the basement membrane. C is what? The podocyte cells. Okay. All right. So let's go to the next question. Fluid is forced into Bowman's capsule by ultrafiltration to form the glomerular filtrate. Describe the role of structure B. The basement membrane acts as a filter. It is actually the molecular filter. So it prevents large molecules that are that are bigger than 68,000 molar mass and above from passing right into the renal into the lumen of Bowman capsule. Okay. It also prevents red blood cells, large proteins, okay, from passing. So acts as a filter, prevent that from happening. So it's the main molecular filter. That's the function of basement membrane B, acts as filter. Okay. So look at this. Say the main component of the glomerular filtrate formed in the Bowman capsule is water. Most of this water is reabsorbed as the filtrate passes along the nephron. 180 of the water forms part of the glomerular filtrate each day. 1.4 dm³ out of 180 water leaves the body in urine. Okay. Calculate the percentage of water in the glomerular filtrate that is being reabsorbed, okay, as the glomerular filtrate passes along the nephron. Show your working. So just the one that is reabsorbed is 180 minus 1.4. So 180 minus 1.4 all over 180 times 100, that's percentage. Okay. So because out of 180 we reabsorb 1.4 is left. So the one that is reabsorbed is this 180, okay, minus 1.4. You know that? All over 180. All over 180. Okay. Okay. Yeah. Times 100%. Times 100%. Yeah. Okay. That's how to calculate it. And what you have is that 99.2. All right, guys. So describe how action. Look at the last question here. Okay. Describe how the action of antidiuretic hormone ADH on the cells of the collecting duct leads to the reabsorption of water in the glomerular filtrate. Okay. So you remember that anti-ADH binds to its receptor on the membrane of collecting duct, and then it then stimulates cell signaling cascade, series of enzyme reaction. So when it binds, all right, what happens? The G protein changes and then activates Of course, the receptor changes and activates G protein. G protein activates adenine cyclase. Adenine cyclase convert ATP to cyclic AMP, which then acts as second messenger and then stimulates signal cascade where vesicles carrying water channels fuse with the membrane. All right, the membrane of collecting duct, and then the membrane becomes more permeable for water to move from glomerular filtrate into the. So look at it. 88 binds to receptor on surface membrane of collecting duct. All right. Okay. Second messenger produced enzyme cascade activation of kinase vesicle. Now these are vesicles carrying aquaporins, so they fuse with the cell surface membrane. So water moves out of collecting duct by osmosis. Okay, that's 7 marks. That's where we stop. Thank you for watching this video. Please share your comments, share this video, let your friends know about it. All right, see you in next series. Bye.